So today I'm going to tell you about our effort as part of the ENCODE consortium for characterizing regulatory elements. So just very briefly introduce why we're doing this. So if you look at the human genome, only 1.5% of the bases in the human genome code for proteins, and the rest is not coding, and particularly among the rest, which is the gray area here, and we're particularly interested in regulatory elements because if you look at all the genome-wide association studies, and if you look at the disease-associated genetic variants in the human population, more than 90% of them lie in the non-coding region of the human genome. And luckily, these variants are highly enriched in regulatory elements. That's why we want to annotate the regulatory elements in the human genome. So by regulatory elements, what do I mean? So I mean promoters, which are immediately upstream of a transcription start site of a gene, and they can be active, repressed, or bivalent. Enhancers that act at a distance, still try to regulate the gene through the promoter. Silencers try to repress it. And very importantly, we have this specific element called insulator elements that are bound by the CTCF protein in the human genome, and they block inappropriate interactions between enhancers and neighboring genes. Because our genome is one big linear string, and it's very important for insulators to come in and divide the genome into neighborhoods. So the goal is so important that there has been a 2-decade effort sponsored by the NIH, NHGRI, to support this ENCODE project with the goal of cataloging all functional elements in the human and some model organism genomes, mainly human and mouse. And the project is very service-oriented, and we want to develop freely available resources for the research community. And here's a picture of the ENCODE consortium before COVID started. And the project has completed earlier this year and has generated a whole bolus of data, hopefully energized a whole field to move forward. So my lab specifically, have been part of the consortium in terms of data analysis. So what kind of data has ENCODE generated in the 2 decades? A lot of it related to chromatin structure, different kinds of chromatin accessibility, transcription factor binding, histone modifications, DNA methylation, RNA transcription, RNA binding protein binding sites. And my lab has been very active in building the ENCODE Encyclopedia, specifically building a registry of candidate Cis-Regulatory Elements and its associated tool called the SCREEN, you might have heard. So how do we define a candidate Cis-Regulatory Elements? So by that, we mean a region in the genome which is roughly a nucleosome sized, like 300 base pairs. Instead of being wrapped around a nucleosome, that region is actually accessible to the binding of regulatory proteins such as transcription factors, these colored blobs. They bind there instead of a nucleosome. And in order to detect cis-regulatory elements, we could use antibodies against each of these transcription factors, pull them down, perform this ChIP-seq experiment. And ENCODE has performed like thousands of these kind of experiments. Alternatively, such regions are often flanked by nucleosomes, which have characteristic tails that are modified covalently, like histone modifications. You can use an antibody toward those tails and pull them down and then map to the genome. So ChIP-seq of histone modifications. And ENCODE has already performed also thousands of such experiments. And finally, you could use the property that I mentioned at the beginning. Such a region is devoid of a nucleosome. So you can try to detect regions that have open chromatin using assays such as DNase or ATAC. And ENCODE also has thousands of such experiments. So here is a characteristic genome browser view of a locus near the SP1 gene. And you see open chromatin regions by DNAs, 2 characteristic histomodifications. K4ME3 is the characteristic modification for promoter, and the K27AC is the characteristic modification for active enhancers. And I mentioned earlier about insulator binding protein, CTCF. So what we do is we combine these epigenetic data to define candidate cis-regulatory elements, these like 300 base pair long regions in the genome that can be candidate insulators, enhancers, promoters. And that is what I mean by cCREs. So ENCODE phase 3 had its group of publications in 2020, and we were lucky enough to be part of this effort to construct such a registry of cCREs. And that was done then by 3 members of my group, putting a lot of effort in collaboration of ENCODE members to develop these cCREs. So Jill Moore, Michael Picaro, and Henry Pratt were students in my lab, and at that time, and they were key contributors of this effort. So here is a sentence from the abstract of the paper. We have developed a registry of nearly 1 million human and then 340,000 mouse cCREs, and covering like 8%, 3.4% of the human and mouse genomes. So that was already published, and we are using these cCREs to do a lot of analyses now. And I want to tell you something new about what we have been doing since then. So still as part of the ENCODE 3 paper, we analyzed the evolutionary conservation of these cCREs. And they're divided into categories here: promoter-like elements, enhancer-like elements, and insulator binding elements in blue here. And you can see they are obviously more conserved than random regions in the genome, which are in gray here. This is using the 100 vertebrates Phyto-P score. However, the conservation on average is true, but if you look at individual elements, a lot of them don't appear to be conserved. And we have cCREs between human and mouse, so we can make the comparison. As you can see that a good 1/3 of the elements are not homologous between human and mouse. So we were wondering, are these low Phyto-P cCREs truly unconserved, or perhaps if we have more genomes, especially mammalian genomes, do they show any conservation in recent evolutionary time? So we were very lucky to be invited to be part of this Zoonomia consortium, led by Shishton Limbaugh et al. And Eleanor Coulson. And we joined this consortium because they sequenced 240 placental mammal genomes. And we were wondering if we could take advantage of these mammalian genome sequences to study the evolutionary conservation of cCREs. So students Greg Andrews, Kylie Fan, and Henry Pratt from my lab led this new analysis I'm about to tell you. So I have already showed you the conservation of vertebrates in these dashed lines. And if we use the 240 placental mammals, so 241 mammals, because dog has 2 genomes in this collection. So you can see the solid lines are from mammal conservation. They are much higher than the vertebrate conservation. Okay, that's a good sign. More genomes in more recent evolutionary history will allow us to discern more conservation. So we delved in and made these heat map. We looked at each cCRE. Remember, cCRE is 300 base pairs long. We counted out of the 300 base pairs, what percentage of these 300 base pairs can be aligned to another mammalian genome. And then we counted the species that will be able to align more than 90% of these positions. Okay, that's the y-axis. And then we counted how many species really cannot align. So less than 10% of the positions are aligned in N2 number of genomes. So you can see all nearly 1 million cCREs fall into this triangle by design, because 90 plus 10 is 100%. So roughly half of the cCREs are highly conserved across the 240 mammals. Okay, and we call them group 1, highly conserved. And very interestingly, group 3, roughly 10% of the cCREs are primate specific. So there are 42 primate genomes among the 240. So they show up as a very bright cluster here. And group 2 is actively evolving. So in comparison to cCREs, these are random genomes. You see random genomic regions. And random genomic regions are much less conserved and much more primate specific. So, okay, with this detailed analysis, we wanted to know how does that compare with the vertebrate analysis. So we went back, used the 100 vertebrates, and performed the same analysis. So the equivalent heat map, as you can see here, most of the density shows up here. Okay, so what that means is most of the cCREs are only conserved up to mammals, and only 4% of the cCREs are conserved beyond the mammals to earlier lineages. Okay, this is a busy plot. I am not going to go into details. We actually cut up the vertebrate heat map into regions, and you can see the highly vertebrate enriched conserved regions, VG0 here. You can map to the mammal triangle, and they show up at this tip. They're just highly, highly conserved. So the conclusion is more than most cCREs are only conserved among mammals, and very few are conserved beyond mammals. That's why when we have more mammalian genomes, we can take advantage of the information and study evolutionary conservation of these cCREs to a greater detail. Okay, we went one step further. We take all the 0.92 million cCREs, and we used their alignment information across the entire 241 genomes. And we used that as features and performed UMAP on all of these cCREs. And you can see the entire 0.92 million is shown in this heat map. And group 1 is highly conserved, and group 3 is primate specific. So you can see these continuum from highly conserved to intermediate conservation, and all the way basically explodes into small clusters. And these are small clusters of primate specific cCREs. And these two plots are based on percentage of the cCREs that are aligned in length. So I'm not going to go into details, but these intermediate cCREs really change, as you can see here. It depends on how much of the length do you require them to be aligned. And that's why we call these actively evolving, because bits and pieces of them are actually highly aligned, but others are not. And then we can layer on this heat map with 240 Philo P score, and you can see group 1 here, only the most extreme tip of the continuum have very high Philo P score. And distance to the nearest transcription start site shows up as ridges in this heat map, corresponds to also high conservation regions of the heat map. And we can ask which cCREs overlap with transposable elements. And you see the conserved tip do not overlap with TEs, while the primate-specific ones, almost all of them overlap with TEs. Okay, so that was cCRE. And as part of ENCODE, we also perform a more detailed analysis of transcription factor binding sites. And we have built this resource called Factorbook, and you might have heard of it. So these are smaller regions between 7 to 20 nucleotide long regions that contain a motif for various transcription factors. And we have built this resource so that we have a collection of these TF binding sites. We call them TF BSs. So we can perform the same kind of evolutionary conservation for TFBSs. So here is the same UMAP presentation for all the sites that are bound by a particular TF, HNF4A, which is the liver-specific TF. And you can see that now these sites are smaller. I mentioned 7 to 20 nucleotides long, rather than cCREs that are 300 base pairs long. So you see the resolution is the HEMAP, the UMAP becomes much more resolved, and the clusters now are very, very sharp. And the individual clusters, I mentioned these blue colored ones, they are primate specific. And you can see that I have labeled 6 of them here as an example. So cluster A here is only in gray apes. Cluster B is in gray apes and old world monkeys. Cluster C is in gray apes and new world monkeys. And D is in gray apes and monkeys. And E is in gray apes and lemur-like monkeys. And then cluster F is in all primates plus 3 related non-primate species at the bottom. Tree shrews and then flying lemur, which is really not a lemur, neither flying. But these 3 are the closest non-primate species. And if you look at the transposomes these clusters overlap with, and roughly A, B, C, D, E, F clusters are ordered in a way that are newer from the newest to the oldest in terms of evolutionary history. And you can see these waves of transposable elements that overlap with is still HNF4A binding sites. Okay, so these are all TF binding sites, but they overlap with various families of TEs. And line 1 is among the youngest TE in our genome. And line 2 is older. You see, line 2 shows up later, peaks later. And ALU is also more recent, and MIR is very old. And LTR is also more recent, and DNA is older. So you can see these different clusters of group 3 elements that are primate specific. A lot of them are derived from multiple waves of transposable elements. Okay, so this is just the entire set of the transcription factor binding sites we have curated, totaling 15.6 million such sites. And you can see group 3, nearly 86% of the group 3 binding sites overlap with transposable elements. Most of them derived from these 3 young families, line 1 ALU and LTR, while group 1 and these other groups of transposable elements do not overlap with these TFBSs do not overlap with transposable elements. Okay, so if you look at another example, I have 367 of them. So here is another example. And you see the fine resolution of these lobes in the UMAP. And I'm just going to jump to right here. And these lobes indicate losses in specific lineages. Like 1 is lost in bats, 2 is lost in New World monkeys, 3 in cetaceans, and 4 in both even-toed ungulates and the cetaceans. Apparently, these whales and the dolphins are highly related to ungulates, and they didn't like the land, so they crawled back to the ocean. And 5, 6, you can see, and all these regions correspond to gains and losses of transcription factor binding sites throughout the mammalian evolution. So, so to take into account these evolutionary speed, the mutational rates, so we actually came up with a Gaussian mixture model to detect constrained elements for each of the transcription factors, because they can be different between different transcription factors, as shown as 2 examples here, YY2 and HNF4A. And then when we fit the Gaussian components, there are 2 means here, mu1 and mu2. And the distance between them tells you these conserved subset, how much more conserved they are compared with the subset that's not conserved, which is more or less the same across different TFs. You can see the conservation here for the conserved subset, and then also the logo. The not constrained subset, they really don't show much conservation. So, so we have this set of constrained elements. Okay, so you can see compared with the group assignment I showed earlier and the philo P score. So basically the most tip of this continuum of UMAP corresponds to the conserved subset of sites that our algorithm detects. So what can we do with these conserved subsets of sites? Okay, so I am going to kind of jump over this slide, just very briefly mention. You see these constrained TFBSs, they don't overlap with transposable elements. And these are the mu1 and mu2 I told you earlier, between the two subsets for each TF, how much is the constrained subset more conserved than the background? And they differ quite a bit. And according to the family, they have a pretty wide distribution. And there is a specific family called Queb, thin finger transcription factors. They show very small subsets for the constrained, and they overlap a lot of transposable elements. So we can go into all the details, but I due to the time constraint, I'm going to scope over this part briefly, and just want to focus on what can we do with these constrained subsets. I'm going to argue that the constrained subsets are more likely to be functional in other genomes. Okay, not in human, because we define them in human, but they're constrained across mammals. They're more likely to be functional in other non-human mammalian genomes. So here is a sanity check. The constrained set is more basically exists. They align in other mammalian genomes. Here we use 4 as an example, while the unconstrained ones are less aligned. And if you look at the logo, if you take each of these into 2 sets and look at the sequence logo, you can see human. These two are similar. But when you go to more and more distant species, the unconstrained ones show a lower information content logo. And these constrained sites, if you take the ChIP-seq data in the respective genomes, the ChIP-seq data in dogs, this is, sorry, chimp, this is a macaque monkey, and then this is dog, this is mouse and rat, and you can see the constrained sites are actually bound, while the unconstrained sites, they are not only not conserved, not aligned, they're also less likely to be bound, especially in the more distant species. And also, the constrained sites in humans are more likely to be protected against the DNAs. So this is DNA's cleavage. Typically, the bound site would leave a footprint with these high flanking regions with cleavage, but the site itself is more protected. So the constrained sites are more protected in 2 different cell lines. Here are 2 different cell lines data, DNAs, than the unconstrained sites at the bottom. So they're more likely to be protected, bound strongly in humans as well. And then finally, we went back to the genome-wide association studies and looked at whether or not the constrained sites are enriched in the human genetic variants that are associated with various human traits. We looked at a panel of 69 diverse traits associated with human phenotypes, different kinds, and see how different partitions of the genome will be enriched in such traits. So the zoonomia defined that they are conserved nucleotides here, and the bars indicate enrichment. And So I just want you to focus on the red bar here, which are the conserved transcription factor binding sites that we have identified in combination with the zoonomia constrained nucleotides. And we add information on top of just the zoonomia list. And additionally, the ones that are constrained by our criteria, not in the zoonomia list, still show enrichment, indicating that our constrained transcription factor binding sites are highly enriched in the variants that drive human traits. And here is an enrichment. So here is our evolutionary constraint percentile by our definition. And you can see that the most constrained subset shows the strongest enrichment. And even the ones that go down to like top 15% still shows higher than the baseline enrichment. And finally, we looked at the traits, two types of traits. One is one group has 16 erythrocyte traits associated with red blood cells, and the other one has 6 immune traits, and then they show corresponding enrichment in a cell type specific manner. So erythrocyte should correspond to K562. So this bar shows the transcription factors that are bound by transcription factors, the sites bound by them in K562 cell lines shows the strongest enrichment, while for the other immune traits, the sites bound by transcription factors in GM12A7A, which is a lymphoblastoid cell line, they showed the strongest enrichment, more enriched than non-hematopoietic cell types at the bottom. So these ones all show less enrichment. So that was basically what I want to tell you today. I have a whole page of conclusions, but just to simplify the situation, I have told you quite a bit about the conservation and the transposable elements and so on. And the take-home message is, if we look at the regulatory elements in the human genome, more than 1/5 of them are innovations in the primate lineage. And the vast majority of these are derived from transposomes. So 1/5 of our regulatory elements are transposome-derived innovations in the primate lineage. However, at the other end of the spectrum, those regulatory elements that are not these 1/5, the other end, the most deeply conserved elements across mammals, which roughly constitute 1/8 of all of our regulatory elements. Those are the ones that show the strongest enrichment in the GWAS, the human trait variants. Those are the regulatory elements that are most likely to regulate diverse human traits. Okay, so that's what I want to tell you today. And I have mentioned the people who did the work, Jill Moore, Michael Picaro, Henry Pratt, Katie Fan, and Greg Andrews, people in my lab. And here are the 2 tools we have built, Screen and the Factorbook, in case you're interested in cCREs and the transcription factor binding sites. And of course, none of these is possible without the very rich data generated by the ENCODE consortium. And we're grateful for ENCODE members for the long 2-decade collaboration and their input to the resources we have built. So that's it. And if you have any questions, I'll be more than happy to answer them.
Thank you so much, Zhiping. That was wonderful. We have time for plenty of questions here. I believe we have one already in the chat here. This is from Imran Shah. Fantastic talk. Are patterns of TF binding sites for superfamilies, for example, nuclear receptors, consistent across mammals?
Yeah, except for the zinc finger family, the crab zinc finger family I mentioned, that they behave very much similar among themselves. The pattern for other families are not nearly as family specific. So they vary quite a bit. And even TFs are paralogs of each other could behave differently in terms of whether or not they are derived from transposable elements. So we have some quite interesting examples.
It's very interesting. Ale?
Thank you. Really, really nice work. So I was just wondering about, because you showed that the constrained regions show more binding in the data set by Ballester. But I was wondering, because there's also a subset of regions that were bound in different sets of species. So I was wondering if that matched the conservation score you had, like if it was conserved or binding with ChIP-seq in the primates. Did you also see that in your constraint measurements?
Yeah, I'm not entirely sure that I understood the gist of what you're asking. So we use this Gaussian mixture model to detect a subset of the sites that are constrained, and the Gaussian mixture uses phyto-P among the 240 placental mammals. So is that what you were asking? What kind of metric do we use?
Yeah. So just to know whether, like, if a site was bound in the 5 different organisms that were using that data set that they showed that were bound in the 5 animals in liver, did you have like a higher score of conservation or not?
Yeah, absolutely. Yes. So I had these example of 4 other species, and we used the ChIP-seq data of HNF4A in the other 4 species. So those are actual binding data. And the constrained sites are mostly bound, while the unconstrained sites are much likely to be bound in these other 4 species.
Okay, because from that work and from other work by Duncan O'Donnell also, there was a clear, so some regions that had high FASCON scores were never bound in the other organisms. So they were bound in dog, but they were never bound in human or vice versa. So I found it really interesting that it seems that your score is able to recapitulate better the cross binding across species. So it's really nice.
Yeah, okay. So if you want to send me an email, I'll be happy to look into it. Thank you so much.
Yep, Shanu?
Nice talk. Yeah, I'm just curious about the constraint elements because if the elements is constrained, is there any preference toward any type of cis-regulatory elements, like in the enhancer or the promoters, things like that? And if so, are they behaving the same across species?
So we have all kinds of elements. We have enhancers, promoters, and a subset of them are more conserved than the rest. And then for the transcription factor binding sites, we looked at 367 TFs. And questions online. What about my answers? Can you hear me well?
Yes, we can hear you.
Okay, good. So we looked at 367 transcription factors, and they behave every each way. So I don't think there is a one size fits all conclusion here, but rather just to continue our effort of, for example, Factorbook, and we really want to be able to show all these data in a TF-specific manner on our Factorbook website. For example, like the UMAP for each TF, we're working on displaying them in an interactive manner, so you can go and look at the individual clusters of sites on our Factorbook. That's work in progress.
I have different company environment among those binding sites. So I'm kind of curious if they function the same, maybe, even the cloning environment is different, the transcription factor itself still can.
You are asking about chromatin environment of the sets?
Inside, so I'm kind of curious about these things. Yeah, thank you.
I have a question for you here myself. So I know you
Yes.
looked at the overlap with the different types of transposable elements, and I might have just missed this, but have you looked at the evolutionary age of the subfamilies within those transposable elements? Do primate-specific sites, are they primate-specific repeats, or do they also overlap recent duplications of ancient repeats?
Yeah, we date. We can try to use a Zoox-Cantor method to date each copy of the transposable elements, and they are actually very interesting. It's like waves of them. Even, for example, like LTR, like those different clusters, they are from different waves of LTR. Some are younger, some are older. So it's really like, we are just bombarded by transposable elements, our genome. It's not like one wave in, it's like many, many waves came in. And then obviously it depends on the age. And as I mentioned, line 1 and LTR and the LU tend to be younger than line 2 and MIR and DNA. And they ease a trend, but under the trend, there are many, many waves of Ts.
Thanks. We have one more question from Sushmita, but just a second, experiment with the echo problem here. If I turn the audio off here at the podium part, does it still come through?
Sorry, no, we can't hear it without your mic on.
Sorry for experimenting at the very end of the conference. Sushmita, ask your question.
No problem. You can come on the mic.
Okay. Oh.
Can you hear me okay? That works. Can the speaker hear me?
Can you hear Sushmita's question? Zhiping, you're muted now. I'm sorry. I don't know why it's that.
I didn't hear it. Maybe Sean, you could repeat that back to me after you have heard it.
Question, but can you hear me?
I don't think she can hear you now at all. She's coming.
We broke something.
Sorry.
Hello, I don't think you see. Oh, there.
I can see you. Hi.
Hi. So I had a clarification and a question. So the clarification was the alignments that you're using is coming from the ensemble compara, or did you do something else?
Oh, okay, I can answer that right away. So the Zoonomia Consortium used a program they developed called the Cactus to do reference-free alignment. So the alignment is not, it's not like a star alignment anchored on the human genome, but it's reference-free. So we just took their multiple genome alignment, and then we have all these corresponding positions across the 241 genomes.
Wow. Is this available? I mean, is
Yes.
this okay? I guess my second question was, maybe it may not make a lot of sense. Do we have time?
That's one, yeah.
Yeah, so when you look for the motifs for different transcription factors, is there a way to, I mean, can we look at it and look at so you look at the motifs, can you look at it and figure out what is a species-specific motif, and how do you do that? So do you get the binding sites and then you basically just get the PWM from there, or is there some more learning happening in terms of getting the species-specific motifs?
Yeah, I'm glad you asked that question. I was trying to fit within my 30 minutes, so I jumped over the details. So we actually built a convolutional neural network model to identify these motif sites in ChIP-seq peaks in human. So for these 367 TFs, we have a whole slew of their ChIP-seq data in humans. And then we build a CNN model to identify the sites in humans. And that's how we have these 15.6 million TFPSs. For other species, except for the 4 I was talking about with ChIP-seq data, and all we showed were the alignment of the human sites across species. Did I answer your question?
Yes, I think so. I can follow up with you. Thank
Okay.
you.
Thank you for your interest.